So welcome to lesson 2. Here we're going to look at B2, organisation. Now, in the last few weeks with this lockdown work, we spent quite a lot of time on the organisation topic, doing recapping, stretch and challenge. So a lot of the components in the paper 1 you should probably do quite well on. You might want to review that work in readiness for that. There's a couple of areas that I didn't really cover that much in the recapping work, which we're going to go through now. And the first one is all about metabolism, and the second one is on food tests. So let's talk about metabolism first of all. So metabolism is, what it means is the sum of all the chemical reactions that take place in the body of an organism. So in animals, that will include all the different types of chemical reaction you can think of: respiration, digestion, protein synthesis, are just examples. So the sum of those is a measure of your metabolic rate. If we're looking at the metabolic rate of plants, it will have those same 3 things, as well as photosynthesis, because that is also a chemical reaction. Okay. Individuals' metabolic rate may vary, and the reason it may vary from person to person could be down to a large number of different factors. Age, gender, the percentage of muscle mass are just a few examples of things that could affect your metabolic rate. Muscle mass, for example, the more muscle you have, the higher your metabolic rate will be, because those muscle cells are going to be more active, they're doing more respiration. As a result, therefore, that kind of sum of all those chemical reactions goes up. As you get older, your metabolic rate generally starts to decrease. Therefore, the sum of those chemical reactions also decreases. Okay, so there is a question on metabolic rate. I'm not going to go into everything there, but that should give you at least a starting point of the basics of the concept to recap and revisit. Let's move on to now looking at food tests and what sort of things we might need to do there. Okay, so with our food tests, there are 4 key nutrient types that we need to investigate. The first one would be glucose, the second is starch, the third is protein, and the fourth is lipids. So what do we want to know from these different tests? Well, the examiner is going to want you to know what chemical reagent is used for each and what a positive result might look like. You might then also have to put that into an experimental context with one of these food tests, and maybe testing different foods, and think about how you might construct that. We'll have a talk about that in a sec. So I'm hoping you already remember the names of the different chemical reagents used, but let's recap it now. So if I'm testing for glucose, I would use Benedict's solution. Another thing you might want to add here is that you have to heat Benedict's to make it to work. All right. If you do it cold, you won't get a reaction. So you've typically got to boil that, or use the word boil or heat. All right. For starch, we would use iodine. For protein, we would use Biuret solution. And then for lipids, there's 2 possible things you can do there. You can use the Sudan 3 test, or the one I like to use is the ethanol test for that. All right. So they're the names of the different reagents. You also need to describe the colour changes we would get. So in the case of glucose and using Benedict's, what you would see here is the solution would go from blue to red. There's a whole range of colours you could get between blue and red, but keep it simple for yourself. Red means that there is glucose present in your food sample. Can't go wrong. Iodine, a little bit more straightforward. It goes from orange to black. So if you've got starch present, it will go black. With the protein, the Biuret test, the solution starts off blue and it should turn purple in colour if proteins are present. Now, depending on which one you do, the Sudan 3 with the lipids, you would get a sort of red ring at the top. And then for the ethanol emulsion test, it would go cloudy. You get a cloudy emulsion. Okay, so they're all the things we want to look at there. They're the basic kind of things. If I was doing an experiment to test for a particular food for any of these nutrients, and I was asked to write a method, I'd first of all consider what my food was. Now, if my food was a solid, then I might need to grind it down, filter it, to get a solution I could test, because it's best to work with foods that are in a liquid form, or I could maybe liquidize it, whatever. If my food sample is already a liquid, I don't have to worry about those stages there. So think about those sorts of things when if you're asked to write a method on how to test a given food. Okay, so that's everything I really wanted to cover with these B2 organisations, since we've spent so much time recapping and revising those areas. So don't forget to look back at things like blood vessels, the blood, etc., to help really reinforce your knowledge with those, because there will be questions in the exam next week on those. So this is Dr. P signing off. Cheers.